flip. Take it. On behalf of the Research Institute for Tropical Medicine and the Treat Asia Network, I'm pleased to present our study entitled Effect of Macrolide Prophylactic Therapy on AIDS-Defining Condition and HIV-related mortality. Mycobacterium avium intracellular complex, or MAC infection, is a complication of advanced AIDS disease. It carries additional morbid effects and is an independent predictor of mortality and shortened survival. With the availability of highly active antiretroviral therapy, or HAART, the incidence of MAC infections has significantly decreased compared to the pre-HAART period. Although epidemiologic data from Asia on MAC infection among HIV-infected patients have been limited, macrolide chemoprophylaxis remains a recommendation of current guidelines. It is recommended to use clarithromycin 500 mg twice daily or azithromycin 1,200 mg weekly as first-line prophylaxis for HIV-infected persons with a CD4 count of less than 50 cells per cubic millimeter. Primary prophylaxis is withheld safely in HIV-infected persons whose CD4 counts have risen to more than 100 cells per cubic millimeter for more than 3 months in response to ART. Along with these recommendations, clinicians must weigh the potential benefits of macrolide prophylaxis against increased pill burden and drug costs, drug interaction and toxicity, and antimicrobial resistance. With all of these factors coupled with the availability of effective antiretroviral therapy and decreased prevalence of MAC infection, we did this analysis to determine whether withholding MAC prophylaxis when it is indicated has significant impact on the occurrence of AIDS-defining conditions and or HIV-associated mortality among patients starting ART in Asia. The data used in this analysis came from the Treat Asia HIV Observational Database, or TEHOD, a multi-center collaborative cohort study involving 20 study sites in the Asia-Pacific region, with the ethical approval being obtained from the IRBs of all participating sites. Patients aged 18 years and above at the time of ART initiation and with CD4 count of less than 50 cells per cubic millimeter were included in the analysis. The data collected included patient demographics, hepatitis B and hepatitis C status, baseline CD4 cell count, HIV viral load, prior AIDS diagnosis, initial ART regimen, and chemoprophylaxis use. The following outcomes were used in the study. Outcome 1 is HIV-associated mortality or AIDS-defining conditions as a combined outcome, and outcome 2 is HIV-associated mortality alone. Censored observations were considered as those individuals who did not experience or develop the outcomes of interest up until 2 years after ART initiation. Patients who were lost to follow-up and whose deaths were not associated with HIV/AIDS were considered as competing risks. Competing risk regression analysis adjusted for the study site was used to evaluate predictors of the two outcomes of interest. Follow-up time was computed for ART start date until the occurrence of AIDS-defining conditions, death, loss of follow-up, or censoring. Follow-up time in all time to event analysis was left censored. Macrolide use, age, sex, mode of HIV exposure, hepatitis C and hepatitis B status, prior AIDS conditions were considered as fixed covariates, while CD4 count, HIV viral load, ART regimen, cotrimoxazole use, and use of other chemoprophylaxis were considered as time-updated covariates. A sensitivity analysis was conducted to assess whether results were consistent in patients with a CD4 of less than 100 cells per cubic millimeter at baseline. The same operational definition and statistical analysis used in the primary analysis were applied. There were 1,345 patients eligible for inclusion in this analysis. 78% of patients were male with a median age of 35 years. Homosexual contact was the most common reported mode of transmission. The median baseline CD4 count was 21 cells per cubic millimeter. And the median HIV viral load was 120,000 copies per ml. 50% of patients had known prior AIDS diagnosis. Of eligible patients, 88% were on ART, 76% received cotrimoxazole prophylaxis, and only 10% of patients were on macrolide prophylaxis. The rest of the characteristics is summarized in this table. Outcome 1 occurred in 99 patients at an incidence rate of 7.35 per 100 patient years. The figure shows the cumulative probabilities of outcome 1 where higher incidences of HIV-associated mortality or AIDS-defining conditions were reported among macrolide users than among non-macrolide users. This may be due to the tendency of clinicians to start chemoprophylaxis when an AIDS-defining condition was diagnosed among these patients. Included in the analysis are patients with severe immunodeficiency, and the possibility of developing AIDS-defining conditions was high. Nonetheless, we found no significant association between the use of macrolide prophylaxis and having the outcome. Risk factors associated with outcome 1 are presented in this table, but simply in the final multivariate model, older age was associated with an increased risk of outcome 1, whereas higher CD4 cell count and use of cotrimoxazole prophylaxis were strongly associated with a lower risk of having outcome 1. For outcome 2, HIV-associated mortality occurred in 46 patients at an incidence rate of 3.14 per 100 patient years. This figure shows higher incidence of HIV-associated mortality among non-macrolide users compared to the macrolide users. This table summarizes the analysis of factors associated with HIV-associated mortality. In the final multivariate model, prior AIDS diagnosis was associated with HIV-associated mortality, while higher CD4 cell counts, cotrimoxazole use, and macrolide use were significantly associated with a lower rate of the outcome. Sensitivity analysis showed consistency of results among those patients with CD4 cell count of less than 100 cells per cubic millimeter, and with lower risk of mortality among those receiving macrolide prophylaxis compared to the non-macrolide users. This analysis showed a significant increase in the risk of HIV-associated mortality among those who were started on macrolide prophylaxis. Although the risk of disseminated MAC infection and mortality is reduced in this group, macrolide prophylaxis is still beneficial against other infections. Azithromycin was reported to provide additional protection against pneumocystis cerevisiae on top of the standard cotrimoxazole prophylaxis. Comparative clinical trials have demonstrated excellent activity of azithromycin against Streptococcus pneumoniae, Haemophilus influenzae, and Mycoplasma pneumoniae, and a significant reduction in the incidence of presumed non-MAC respiratory infections, specifically pneumonia and sinusitis. Clarithromycin prophylaxis reduces the risk of bacterial respiratory tract and soft tissue infection, thereby significantly reducing mortality among HIV patients. In conclusion, macrolide prophylaxis is associated with significantly improved survival among Asian HIV-infected patients with very low CD4 cell counts. The benefits of giving macrolide prophylaxis remain despite the availability of effective ART and the decreased prevalence of MAC infection. This study suggests the need to expand macrolide prophylaxis usage and coverage among HIV-infected patients in the Asia-Pacific region. Thank you.
Thank you, sir. And we now have the questions from our judges.
Thank you very much, Dr. Pasayan, for your presentation. For me po, kasi medyo medical and scientific presentation. So, I will be giving a parang very general question lang po na of importance to us. So, given the findings of your study, can you please at least provide me one policy recommendation that we can actually provide to policy makers para may present to sa kanila? Thank you.
It's not specifically sort of policy, but a guideline changing recommendation, because our current guidelines is giving azithromycin as prophylaxis for people living with HIV who are who has severe immunodeficiency, and this is indicated only for MAC prophylaxis. But from this study, since the MAC prevalence in the Asia-Pacific region is very low, the indication for giving MAC is now different. It's to prevent HIV-associated mortality and not specifically for MAC prophylaxis per se.
Thank you. Good afternoon. Thank you very much, Dr. Pasayan, for your presentation. I have a couple of questions. Some of these relate first with, I guess, more curiosity. I know that this is just a snippet of a bigger study that's being conducted. I think it's ongoing, no? This DOH, as it's been called. So just for my understanding, no, in terms of the methodology, this portion on the effectiveness of macrolide prophylaxis is really only a dimension. How does this work in terms of the general component of the bigger study? Can you explain that a bit?
The DOH map is DOH is a big Asia HIV observation.
The database,
It's a
isn't it?
database
Yeah.
Supported by AMPAR, American Foundation for
All
AIDS
of these
Research.
institutions
All of
that
this.
are part of
Yes.
it.
And the Philippines is one of the study sites,
Okay.
together with 20 other study sites in the Asia Pacific region,
Okay.
including Australia. And our ITM is the source of the Philippine cohort.
All right.
Now, we collect data from patients and transfer it to a bigger database, which is based in Australia. And for whatever, let's say, HIV-related concerns or study research, can get or extract data from this database, anything that is related to your topic or to your.
Okay. Now, in relation to this, my understanding is, so this is a sort of like a retrospective
No,
cohort. Meaning, all of these are just going on, and is it correct? You have to educate this non-clinical mind. All of these are going on, and is it correct, therefore, to say that the treatment guideline in relation to HIV and AIDS defining conditions differ in the aspect of the use of macrolide prophylaxis? Is that correct?
Yes.
And therefore, in your database, you can extract those that were given and those that were not given.
Yes, ma'am.
Okay. Yeah, that's important to clarify, because I guess for sitting through so many proposals in an institutional ethical review board, you kind of are like, did they do this and deprive the others? That kind of question comes to mind. So it's clear that you did not, this study, or at least this snippet of the study, did not have anything to do with determining that this group will receive this other
No,
group.
no.
Okay. Now, having said that, my last question can you please pull up your slide that has the balance? There. Sorry, the balance, there. I know that this is the background, but from an application point of view, I wanna connect to what Pio was saying. The applicability, you're saying to change the guidelines.
Yes.
I put myself now in the seat of a policymaker or that which creates policy. You gotta tell me this. You gotta tell me how the benefits will far outweigh
Yes.
cost, perhaps.
Yes.
Interactions and all of these other things. I know that it may not be totally fair to ask you because that may not be within the purview, but if this is the background and you're pointing towards eventually leading to this, can you kind of just give us some idea to convince a policymaker that this is worth pursuing to actually change the guideline?
Because ma'am, if we would stick to the guideline per se, the only reason why macrolide is being given to patients is for a certain prophylaxis against a certain bacteria, a certain AIDS-defining condition. Well, in the Philippines, the problem is always the cost. It's very, very expensive. And if this is a prophylaxis that the patient might take at least a year. Now, if we are going to just say the reason for giving this prophylaxis is for Mycobacterium avium complex, we don't have the latest data, even in Asia, regarding the prevalence of Mycobacterium avium complex. And studies in the United States and even Europe show that the prevalence is now going down because of the antiretroviral drug. So if you will just base the reason for giving prophylaxis is the MAP, then this will just be going to tip down to the left. But from the study, it's not actually the MAC that is the benefits not actually just for the MAC prophylaxis, but also for the HIV associated mortality due to other infections, which azithromycin or clarithromycin can also prevent.
Let me restate what you said. That therefore, you're saying that the benefits of macrolide prophylaxis will far outweigh the disadvantages attributed to burden, pill burden, and
Yes.
cost, because they don't only prevent, they encompass a spectrum
Yes.
that of effects, of beneficial effects.
Not just a
Now,
single-
having said that, since I missed out some recommendations and you just simply concluded this paper with a statement, you might want to tell us right now what then will be a recommendation, be it, again with the end in view of achieving an objective of policy change in terms of guideline. Maybe that will be my last question. You might want to give us that recommendation.
The current recommendation in the local guidelines for macrolide prophylaxis to give it if your CD4 is less than 50. Now, when we did a sensitivity analysis, including those population with CD4 of even less than 100, the benefit of macrolide prophylaxis is still there. So one change actually that we could recommend is to extend the coverage of macrolide to those patients not only with CD4 of less than 50, but even to a higher range of less than 100. Because the benefit is still there. When you reach, say, 100 of CD4 of 100, the tendency or the probability that you'll get MAC infection becomes less. But still, the benefit that a patient could get from macrolide as prophylaxis against other infections is still there because the patient is still immunocompromised.
So, sorry, I can't let this go. So, what would then the Health Policy Development and Planning Bureau Research Division offer as a potential further research? I'd have thought that you stated it in a very clinician scientific way. And I'd have thought to prove the case that macrolide prophylaxis is really important and must be part of the standard guideline or protocol, would there be a benefit perhaps of, or a possibility even, of a more cost-benefit directed kind of study? Is that feasible? Is that even possible? If you say, can that be done? Can you see your way through doing something similar with the information that is at hand from the TEHO database, perhaps?
Well, maybe not from the TEHO database because the Philippines only contributes 200 patients in that database. But if you're going to base it locally, yes, there is, ma'am. We can actually compare the effectivity of giving this prophylaxis in a resource-limited setting like the Philippines, and where where Mycobacterium avium complex is really not common, not as common as tuberculosis, and perhaps also study the possibility that we might not be dealing with TB alone, but the relative, which is Mycobacterium avium complex.
Thank you very much.
Thank you and good afternoon.